In late March of 1884, during a brief trip to the Villa Nevada in Cannes, France, Prince Leopold, the first Duke of Albany, was visiting the nearby yacht club when he suddenly slipped on the interior tiled floor, causing some tissue damage to his knee and blunt force trauma to his head. He was later taken to a bed to recover from the sudden injuries he had sustained in the club, but in less than 12 hours later, he was pronounced dead from a cerebral hemorrhage, as the blood vessels in his head had suddenly exploded. What's interesting about his death is not only the sudden shift from partygoer to deceased individual in less than half a day, but for the majority of his life, Prince Leopold was under regular supervision by a team of physicians and had dealt with a variety of physical injuries and symptoms throughout his entire life. And oddly enough, the fact that he lived to the ripe old age of 30 was actually noted as a medical marvel at the time. This same disease also went on to affect 8 other individuals in his extended family, with 8 additional members being genetic carriers of said disease, ravaging its way through the royal family lineage to the point where the two were then intertwined into history forever. So, what on earth was going on, and how did this affect 17 members of the royal family across 7 generations and 120 plus years? What was this death sentence? The individuals in the royal family suffered from a genetically inherited disease known as hemophilia B. Hemophilia is a combination of the Greek words for blood and love, which is another way of saying that people with this disease love to bleed or can't stop bleeding. And that's horrible. Normally, when you sustain an injury to your blood vessels, there's a series of steps that your body takes to ensure that the bleeding stops and the injury is repaired properly. This is done in 2 major steps, primary and secondary hemostasis. The initial response to vascular injury involves reducing blood flow to the affected area, so primary hemostasis begins with vasoconstriction, which is a rapid narrowing of the blood vessel triggered by damage to the endothelium and serves to minimize blood loss. Following this, platelets are recruited to the site of injury where they adhere to the exposed surface and to one another, forming this temporary patch known as a platelet plug. The thing to note here is that even though a ton of platelets are clogging up the injury, there's still leakage and blood loss here, and why part 2 is so essential. The reason why part 2, or secondary hemostasis, is so important is that it's to produce this wiry looking substance called fibrin. This material is incredibly important in the tissue repair process, and it creates this mesh-like network in the injury and stabilizes the clot to keep the cell components on the proper side. So the production of fibrin is initiated through 2 distinct pathways, the intrinsic and extrinsic pathways, each activated by different mechanisms in the body and operate independently at first. However, despite their separate origins, both pathways eventually converge into a unified sequence known as the common pathway, which results in the formation of fibrin for proper clotting. But if you look at these pathways, you might notice that fibrin is the end product of all of these steps. And this is why hemophilia is so deadly. Do you see all of these steps in the cascade? The royal family had hemophilia B, and therefore missing clotting factor 9, which means that this entire side of the intrinsic pathway is basically screwed. This coagulation cascade process needs every step to work to form a proper solid clot. Having an issue with one of the clotting factors would affect how well the clot forms on the injury, since the next step in the cascade would be impeded by the previous clotting factor not working or missing. And of course, this had some devastating effects on those who had the disease. Of the 9 members of the royal family who were fully affected by hemophilia B, only 4 lived beyond the age of 30, while the major succumbed to acute trauma during infancy or adolescence. And it kind of makes sense. Cuts, bumps, and bruises are rather trivial injuries to most of us who are free from blood disorders, but imagine living a life with the knowledge that even the simplest interactions in life can kill you. A single mutation managed to inflict such disproportionate devastation upon one specific royal lineage? To answer this question, we can start by looking at one key element that is at the core of so many debilitating illnesses across humanity's history, and that's genetic inheritance. Human beings possess a total of 46 chromosomes, organized into 23 complementary pairs, one set inherited from each parent. Among these pairs are the sex chromosomes, with females carrying 2 X chromosomes, designated as XX, while males possess 1 X and 1 Y chromosome, or XY. And this specific distinction is at the core of why hemophilia is transmitted so predictably. Some genes only live on certain chromosomes, and the clotting protein instructions that prevent illnesses like hemophilia live exclusively on the X chromosome, giving this phenomenon the characteristics to be an X-linked trait. Hemophilia is also considered to be X-linked recessive, with the recessive portion of this meaning that if on your sex chromosomes you have two different types of gene copies or alleles, like one wild type or one mutation, the wild type or normal version will take over as the dominant trait between the pair. But in the case of males, because there's only one X chromosome present, so you're kind of just stuck with whatever you inherited. So let's start with Queen Victoria. Because she's biologically female, she holds the genetic makeup for hemophilia but doesn't have the disease herself, thanks to one of her X chromosomes being the wild type variation. And so she's designated as a carrier. Her husband, on the other hand, Prince Albert, had no markers for hemophilia B, so he's designated as an unaffected. In this specific configuration, with a carrier mother and unaffected father, there are 4 possible outcomes, each with a roughly 25% chance of occurring in their biological children. There's a 25% chance of an unaffected son, 25% chance of an unaffected daughter, a 25% chance of an affected son, and a 25% chance of a carrier daughter. In simpler terms, 50% of her sons will be affected with hemophilia B, and 50% of her daughters will be carriers for the disease. So if we know that hemophilia B is inherited through a sex-linked recessive pattern, and research shows that none of her parents had or even carried the genetic makeup for this disease, how did it end up starting with Queen Victoria in the first place? One of the more interesting theories surrounding the British royal family is the speculation that Queen Victoria's father may not actually have been her biological parent. The controversy that surrounded the origins of hemophilia in the British royal family took a provocative turn back in 2003 when biographer A.N. Wilson put out a bold and rather contentious theory regarding Queen Victoria's true parentage. He suggested that Victoria was in fact not the biological daughter of Edward, the Duke of Kent, but rather the child of an illicit relationship between her mother and an Irish soldier named John Conroy. Wilson's theory sought to explain the sudden appearance of hemophilia into the royal bloodline, a genetic disorder previously unseen in the family, but then retracted his statements, acknowledging that Victoria was indeed born of a legitimate union, citing some familial resemblances and the absence of certain genetic conditions that were previously thought to have supported his original claim. But the most likely reason why hemophilia specifically started with Queen Victoria was actually a rather surprising one, in the sense that it just sort of spontaneously happened. There's a specific kind of mutation called a de novo mutation, which is used to explain any sort of genetic alteration that presents itself for the first time in a family member. And this all seems incredibly random, but actually in around a third of patients, hemophilia B pops up due to the onset of a de novo mutation in a family previously unaffected by the disease. And that's really it. A single and spontaneous genetic mutation in just one individual caused absolute devastation across multiple generations. And given how things started, it could just as likely happen again.